Good morning, 8th class students. Welcome to virtual classes. How are you all? How did you write your exams, FA1 exams? And have you participated in Batkama celebrations and Dasra? Yes, belated happy Dasra to you all students. So once we have completed recall our topics, we have completed our second lesson. And have you written your notes? Yes or no? Every student, please type your name and branch name. Okay, students? Yes. Yes, Prahashita, Prasanna, Satvik. Okay. So today we are discussing about the story of microorganisms. Is this the same lesson name in your textbooks also? Story of microorganisms or microorganisms friends and foe. What is the lesson name? Same lesson name? Students, once you check your textbook, yes, let us learn the third lesson. So, what is the story of microorganisms? So, microorganisms are very small in nature. So, in your home, your mother prepares curd by how? By mixing milk, by mixing what? Some drops of curd in the lukewarm milk. What? Some drops of curd in lukewarm milk. In the lukewarm milk. Isn't it, children? And next, as soon as you get up, your mouth will produce a bad smell. And your mother will prepare a dosha batter and idli batter, no? She will keep or she will rest that batter for half day. The world of microorganisms. What is the lesson name? What is the third lesson name? 8th class students. Story of microorganisms or world of microorganisms? In your textbook it was given, no? The world of microorganisms. Part 1. Yes. The names. Actually, my textbook was the publication is the last publication is 2014, my textbook. So that is why the name is different. That's why I asked you the world of microorganisms part 1. First of all, the microorganism was named, the lesson name was named as the story of microorganisms. Later after 2-3 years, it was published as microorganisms friends and foes. Friends means they are like friends. Foe means enemies. F-O-E, foe means enemies. So some microorganisms are like our friends useful and some are not useful, no? So like that. But now we are having the textbook with what? Third lesson? The world of microorganisms. Whatever the lesson name, the inner content is same only. So here when we get up in the early morning, our mouth will be with a bad smell. Isn't it, children? So we can write like this also, world of microorganisms. Yes. Feeling happy now? Yes. Students, listen here. If we want to read about microorganisms like air, no? Air is present everywhere. Air. Can you catch air? No. So like that, everywhere is filled with microorganisms. Micro means small, very, very, very minute. So what will come under microorganisms means bacteria, algae, fungi. Next, now we are facing a big dreadful pandemic situation because of coronavirus, COVID-19, that belongs to the family virus. This is also a microorganism. Virus. Bacteria, algae, fungi, viruses, this all comes under microorganisms. So now you feel it is okay to study about this? No. You are studying about now streaming, the worldwide streaming coronavirus. Isn't it, students? So what is the microorganisms? Who observed microorganisms? Who named microorganisms? How is it possible to observe a microorganisms? This all we will study in the microorganisms. So your mother will prepare curd, no? In that lukewarm milk she will add a small drops of curd into the lukewarm milk. That curd consists of microorganisms. The curd that we are eating is nothing but microorganisms. Isn't it, students? You are eating curd now? Yes, the curd is nothing but microorganisms, that is bacteria. Okay. Lactobacillus. What is the name of the bacteria present in milk and curd? Lactobacillus. What is the children? Lactobacillus. They are rod-like structures. If you observe it under the microscope, you will observe like these structures are present. Those are nothing but bacteria. They are rod-like structures. They are present in the milk and curd. Milk and curd. And next, your mother will prepare batter, no? Idli batter, dosha batter. After some time, she will rest the batter for night. She will grind the batter in the evening and she will keep rest it for next day morning to prepare dosha and idli. Once you check the batter before after grinding and in the early morning you observe what happens, it becomes bulged. Some smell will come if you open not clear board. Is it okay now? Rajkumari. Yes, clear only. That may be yes problem. Yes, idli and dosha preparation. So what your mother makes a batter up to here and close it with a lid. In the early morning, what happens? This become more mass. This is because here yeast is present. Okay, children? Yeast. So, as soon as you open the lid, the lid of the vessel, you will see some smell because their bacteria is produced. That bacteria is only fermenting the idli batter or dosha batter. Why became the more mass? That is what I am saying. Why it is becoming more mass? Because yeast, the bacteria, it is getting divide on the whole night and it becomes big. Which bacteria is more dangerous? We will discuss that one. Now it is only some description. Yeast is a bacteria. Yes, it is a bacteria. It goes on. This yeast will be like this in structures. Yeast. Okay, students? Yeast. So this yeast acts on the batter the whole night. It divides, it acts on the batter and it becomes to ferment. What it does, it makes fermentation. In that fermentation, they release CO2 gas. Okay, 8th class students, are you following? Yes. So what happened? We prepare idli batter, no? Or dosha batter, no? You ask your mother, can we get nice prepared idlis if you won't ferment? You ask your mother, Mama, we have grinded batter. We have prepared batter. Can we make dosha? Can we get correct doshas? Can we get correct idlis? Means what she will say? No, you have to keep this batter without disturbing for some hours, she will say. What is the reason? Means the bacteria acts, the yeast bacteria acts on the batter and it ferments the batter. It goes on dividing and it ferments the batter, and during that fermentation CO2 is released. What happens? CO2 is released. That is why once you open the lid of the batter, no, you feel some smell, some foul smell you will notice. Isn't it, students? Yes or no? So what do? You now after the completion of this session, no, you ask your mother to prepare the dosha batter or idli batter. Tomorrow you notice in the early morning. So you say you are feeling. Until you, as soon as you open the lid, no, you feel some smell it comes because this yeast release CO2 gas during fermentation. During fermentation. This is about what the useful of microorganism, that is yeast. Okay, students? So, 400 years before, 400 years before, they don't know about these microorganisms. Means something was there, maybe a superpower or God is doing this type of things they have thought 400 years before. We prepare sambar or food, no? Next day it gets spoiled and we sleep during night, and as soon as you get up in the morning, a foul smell comes from our mouth. And curd, only during the night only we are adding some drops of curd. We are adding some drops of curd on the lukewarm water, and in the early morning, what? Not understanding. What you are not understanding, Rajkumari? Yes, listen here, students. So, we prepare the food. The food gets spoiled in the next day, and you are adding to the milk what? Some drops of curd. In the morning itself, you will observe the milk is totally turned to curd. The The milk is totally turned to curd. So what happens during this process? They don't, 400 years before, many years before, they don't know answers for these questions. They thought something was happening. But after 400 years, means during 1674, during 1674, a scientist named Antony van Leeuwenhoek in 1674, Antony van Leeuwenhoek, he collected stream water or running water or lake water, whatever it is, stream water or lake water or running water. He took the stream water, running water sample and he observed it under his own prepared microscope. This is the type of microscope used by Antony van Leeuwenhoek during 1674. Not understanding. Okay students, you follow, you will get better understanding. So what happened in the 1674? Before 1674, no one knows about microorganisms, students. But after 1674, it was possible. Why? Because Antony van Leeuwenhoek, a scientist, a human being. So Antony van Leeuwenhoek in 1674, he used this type of microscope. During those days, there is no microscope like we are using nowadays. So this type of microscope he used for what? For observing stream water. He collected stream water and observed it under the microscope. Antony van Leeuwenhoek. Okay students? Antony van Leeuwenhoek, what he has done in 1674? We are living in 2020, no? We are living in 2020, 1674, 18, 19, 20. So 400 years, no? Nearly. So before that, in 1674, Antony van Leeuwenhoek, he collected stream water or running water or lake water and he observed it under the microscope. This was the microscope that was used by Antony van Leeuwenhoek. You observe, students, before this 2020, we have used some mobile phones only for talking, but after they have used for video calls. Now we are talking in group video calls. Isn't it, students? Like that. Many modification happens. In 1674, it was not technologized and digitalized version. At that time only, Antony van Leeuwenhoek, he prepared his own microscope and observed the stream water. And in the stream water, he observed small, they are moving like this in the water. In a drop of water, he observed small, they are moving. Small animals, they are moving. Okay, and he named them as animal clues. Animal clues. C-L-U-E-S. Okay, students? Animal clues. Okay. At that time, they were not named as microorganisms. He named them as animal clues. And this made to study various type of microorganisms. This was named to study different types of microorganisms. So that is why Antony van Leeuwenhoek is considered as father of microbiology. Father of microbiology. Yes, students, listen carefully. Our lesson is world of microorganisms. So we want to know who discovered microorganisms, how they were discovered, that we have to study. So what is that? In 1674, Antony van Leeuwenhoek, he observed the lake. he observed the lake water, and in the microscope was prepared by him, and he observed somewhat some things are moving in the animal, in the sample, the water sample, and named it as animal clues. And what he named it as animal clues. That animal clues are nothing but now we call them as microorganisms. He was the first person to discover microorganisms. That is why we call Anthony van Leeuwenhoek as the father of microbiology. Father of microbiology. Okay, this is about the Anthony van Leeuwenhoek, that history of microorganisms. So here students, animal clues or animal clues. In some textbooks they are given us clues. In some textbooks they are given us clues. Okay, no need to worry about that. So Anthony van Leeuwenhoek is a fabric merchant. So he prepared his own microscope and observed it under the microscope and named that microorganisms as animal clues, and later on they were named as bacteria, and after that they were named as microorganisms. And let us see some structures of bacteria. These are yeast. Students, these are yeast. Yeast generally is used in the preparation of cakes. Okay students, cakes, idli batter, dosha batter, yeast. Okay, next one is rod shaped structures. Lactobacillus. What are those? Lactobacillus. E. coli. Okay students, E. coli. Penicillium. Okay students, what is this? Penicillium. This is what? Rhizopus. This is fungi. Yes, this is fungi. So this rhizopus you will observe in the bread. Okay, this rhizopus is present on the bread. Okay, what you should do, you take bread and you sprinkle some water on the bread and keep it aside. After 1 day or 2 days, you will see black colored structures that is visible, and you can see on wet coconut also. also. Wet coconut and some vegetables also you will find this color. If you clearly take into sharp vision and observe now, you will see this type of structures. Easily you will see this type of structures. Okay, students? This is aspergillus. Aspergillus. Amoeba. Okay. Paramecium. Okay, students. Paramecium. Euglena. Okay. Euglena. Chlamydomonas. These all are microorganisms only, students. All different structures. You see how they are different structures. Single cell different structures. This one is spirulina. This one is spirulina. We are eating spirulina, students. In the out of states, like English states, yes, aspergillus is dangerous. Once you observe penicillium. So, have you ever heard about penicillium injection? Vaccine, penicillin injection. From this only we are preparing penicillin injection. Spirulina. This spirulina is single cell protein, students. For your better information I am saying, spirulina is single cell protein. Proteins is present in our diet, no? Proteins. Yes. This one spirulina, this we are consuming. Okay, we are eating this spirulina. If you are suffering with protein deficiency, okay, so these are the many types. Next one is this type of structure, Spirogyra. What is that? A Spirogyra. Spirogyra. Verticellaster. What is that known as? Verticellaster. How it is? See, verticellaster, how it is? It is like a hibiscus flower, no? Verticellaster. Which is harmful in this? Nothing is harmful. Some fungi are harmful, bacteria this. So here yeast. Yeast is present in batter. We have explained and we are using in the cakes preparation. And lactobacillus that we are eating in the curd. And E. coli, no? E. coli is used to treat some sugar diseases, diabetic diseases. Then we will learn these are the types of bacteria. We will learn which one of this is useful and which one is harmful next in the next topic. So here one topic is there. Activity is there. What? Collect some pond water or water from any tank in your surroundings. Okay, students. And take some greenish scrapings from the side of the tank. Anyway, in the water tank you will see some green color. What we call in Telugu is pakuru. So you take that one and take 1-2 drops of water and observe through the microscope. Draw rough sketches in your notebook of what you observe. Compare it with the figures given above for observation. So generally what are the green color structures that you are observing? No? Chlamydomonas, Spirogyra, Verticilliaster, Spirulina. These all are present in the water scrapings, green color scrapings. And can you name the organisms which you have observed through the microscope? You have to observe it under the microscope and you have to compare with this. Which one you are noticing? And observing fungi. This is about observing what algae, and the next one is observing fungi. So usually after the rainy season, you might have seen some small umbrella like growths over rotten materials of dumped waste. Rotten means what? Kullipoyina, damaged one. In the dumped waste between the grasses in fields and edges of wet rotten wooden planks. Yes, everyone, do you know about mushrooms? It is also fungi, students. Mushrooms. It grows in that cow dung or buffalo dung. It is waste, we think, no? So during rain, yes, ma'am, I don't have microscope. Everyone is don't having microscope. Just now you are you are learning from your home, and we are in the school. So don't worry about that. As soon as school started, here only 8th class, no? Next year you have chance, and that last of this academic year you may be allowed to come to school. At that time you can watch everything clearly. Okay. So here on these areas you can find some fungi. Now let us look at them more closely by the following activity. So take some rotten part of the vegetable and a black spoiled part of the bread or coconut. Yes, ma'am, I know mushrooms. Some people will eat. Not only some people, you can eat mushrooms. Everyone can eat mushrooms. It provides vitamin D and protein. That is good for bone development and muscle development. You can take mushrooms. Being fungi also, it is edible. So useful microorganism, no? Yes. Put a drop of water, place a cover slip on it and observe it under the microscope. So photographs, you know, curd. And if you want to observe lactobacillus, it is easy to observe in the buttermilk or curd or tongue. On the tongue also, you can see lactobacillus bacteria. And the next one is we can also find them in the soil, over bark of trees, over our skin, in our armpits and many other areas. But they are not visible to the unaided eye. We can't see directly with our eye. We need microscope in our. But mushrooms are poisonous. Yes, there are 2 types of mushrooms, students. Yes, very good thing. There are 2 types of mushrooms. Some are edible mushrooms, some are toxic mushrooms. Some are toxic mushrooms. If you see a movie by name, I don't get that movie name. If we eat some mushrooms, we fall down. And if we eat some toxic mushrooms, we may get some diseases or we may die. In that also many types of mushrooms are present. Some are edible, some are not edible, some are poisonous. Okay, it causes anesthesia. If you eat some mushrooms, we may fall down. In Telugu, what is called mushrooms? Puttagodulu. So a movie name, I'm am not getting that name of that movie. In that movie they used to be going in the forest. There they find some mushrooms and they will pluck and eat. So they get some anesthesia and they feel as they are injected with anesthesia and they will get fall down. So some are edible and some are not. And here, so here one preparation is there. We have to take one drop of buttermilk or curd and keep on the slide, and on that you have to keep crystal violet solution. In the second lesson we have discussed about preparation of preparation of slide. Preparation of slide, isn't it, students? In the preparation of slide, you will be what you are adding: Japanese green solution, methylene blue solution, saffron in, and here what we are adding: crystal violet, because they are lactobacillus bacteria, no? So only the bacteria will be visible in violet color, remaining you will see as white color only. Okay, so here draw rough sketches in your notebook of what you have observed. So no need of observing it under the microscope, curd or buttermilk. You keep that curd for 3 to 4 days. You can notice small, they are moving like that. Okay. So this is about today's topic. Tomorrow we will continue the lesson. So many the thing I have to say is microorganisms are present everywhere. Some will be present on the surface area, some will be present on the in the air, some will be present in our body, also in the digestion. So microorganisms are present in our body, inside the stomach. That microorganisms help in the digestion. Okay, students. Next, in the what I will say an example. Cattle are there, no, buffaloes, cows. They are eating what, grass. Can we eat grass? If we eat also, we can eat. You will say blindly, why can't we eat? We can eat. But what happens? They don't get digested in our stomach. But it gets digested in the stomach of what? Buffaloes. Because in the buffaloes and cattle, like cows, buffaloes, goats and sheep, no, in that some bacteria is present that digest the grass. In our stomach, that bacteria is absent. So you can note that from this statement you can conclude that microorganisms are useful or not? Useful. Okay, so some bacteria we are eating in the form of curd. We are eating bacteria in the form of curd. Is it useful? Yes. If that bacteria is not present, can we turn the milk to curd? No. So you will see in media that we, in place of curd also we can add badam juice or badams to prepare curd. If we use badams also, microorganisms are present everywhere, no? They come on sit in the milk, and already some type of bacteria is present in the milk, and they undergo division. And they become into curd. So microorganisms are present everywhere, students. Okay, students? So based on their function, we divide the microorganisms into useful microorganisms and harmful microorganisms and symbiotic microorganisms. This is about today's topic. We will continue in the tomorrow's class. Any doubts, students? No doubts, no? So meet you in the tomorrow's class. Stay home. Have a happy learning.